Welcome to AI Explains. Today we're diving into some research from the University of Chicago that honestly sounds like it's straight out of a sci-fi novel.
It really does. We're talking about merging two worlds that just, they don't usually play well together.
Not at all.
Yeah.
You have the cold, super delicate world of quantum physics.
And then you have the warm, wet, messy world of biology.
And usually if you try to put a quantum sensor inside a living cell, you don't get a living cell anymore.
You get a tiny ice cube. Yeah, the whole thing just freezes and dies.
Exactly. But these researchers found a kind of loophole. They didn't build a machine to shove into a cell.
They found a part of the cell that was, it turns out, already a tiny quantum machine.
And the wildest part is, biologists use this thing every single day. It was hiding in plain sight.
It was. The protein is called enhanced yellow fluorescent protein, or EYFP for short.
Which is basically the standard lab highlighter, right?
Yeah.
If you're a biologist and you want to see, say, where a nucleus is,
You tag it with this protein, you shine a light on it, and it glows yellow. It's just a flashlight.
just a flashlight. But this team in Chicago, they realize this flashlight has a kind of hidden switch.
Mm-hmm. It turns out EYFP has a property called spin.
Okay, so let's break that down. For like a 12-year-old, what is spin?
Imagine a tiny subatomic compass needle that's buried deep inside that glowing protein.
So every single one of these little flashlight proteins has a tiny compass inside it. But, I mean, a compass is useless if you can't read it.
Precisely. And that is where the breakthrough is. They found that if they hit this protein with very specific pulses of laser light.
A blue one and an infrared one I saw.
That's right. With those, they could basically grab that compass needle and force it to point in a specific direction. They call it initialization.
So they align the needle. What's next?
Then they hit it with microwaves. Now, if there are any magnetic fields nearby, that little compass needle starts to wobble.
And here comes the magic trick, I assume.
Here it is. When that needle wobbles, the brightness of the yellow glow actually changes.
Oh, wow. So that's the aha moment. It's not just glowing anymore.
No, it's not.
The light is flickering a code back to you, a message based on the magnetic fields it's feeling.
You've turned a simple highlighter into a quantum sensor.
And that solves the biggest problem in this field, which used to be hardware. Before this, they were using, what, microscopic diamonds?
Yeah, these things called NV centers in diamonds. And you can imagine trying to shove a tiny shard of diamond into a soft, squishy human cell.
It's like putting a rock inside a water balloon. You're going to pop it.
It's invasive, it's clunky, but since EYFP is a protein, you don't inject anything.
Right.
You just give the cell the DNA instructions, and the cell builds the sensor all by itself.
So it's like you're downloading an app for the cell instead of installing new hardware.
Exactly, it's genetically encodable.
So they have this incredible theory. Did they actually get it to work inside a living thing?
They did. They ran the experiment inside human kidney cells and also in E. coli bacteria.
I have to assume the signal was messy, though. A cell is chaotic. There's stuff bumping around, chemical reactions firing off everywhere. That's a nightmare for anything quantum.
It is noisy, for sure. When they froze the cells down to liquid nitrogen temperatures, the signal was, beautiful, really crisp and strong.
But our kidneys don't work very well when they're frozen solid.
Right. And this is the crucial part. They proved they could still detect the magnetic fields at room temperature.
Even though the signal was a bit fainter.
A bit fainter, but it was there. And the fact that it works at all is huge. It means we could eventually have these sensors working inside a patient.
Sending out data in real time about what's happening at a molecular level.
We could watch a drug bind to a protein or see a neuron fire with, quantum precision.
It's a whole new way of seeing. We aren't forcing our technology into biology anymore.
We're upgrading the biology to be the technology.
And it leaves you with this wild thought. We have been staring at these glowing proteins for decades.
For a very long time, yeah.
Completely oblivious that they were capable of quantum mechanics this whole time.
It was hiding in plain sight. Makes you wonder what else we're missing.
If a standard lab protein is secretly a quantum sensor, what other superpowers are hiding in the biology we look at every day?
That's the question that's going to drive the next 10 years of this research.